Следующий докладчик Мануэль Йепес, please.
Okay. Good afternoon, everyone. Thanks for having me here today. It's a pleasure and an honor to be here. I'm going to talk to you about neural repair in the ischemic brain. I will not talk to you about the stem cells. I'm going to talk to you about something different, how to move something from the bench to the bedside. That's pretty much what I'm going to do. And I plan to do that over 35 minutes. And my plan is, number one, to talk to you about the facts, why this is important. Then I'm going to introduce the system I'm working with. That's going to be followed by what is that system doing in the ischemic brain. I'm going to make a case for that system to promote recovery. And finally, I'm going to finalize with my concluding remarks over 35 minutes or so. The facts. Why this is important? If you look at the last 14 to 17 years, there's been a 68% increase all over the world in the number of people experiencing an acute ischemic stroke. Nowadays, we can do a number of things. These people survive because we can pull the clot out, we can lyse the clot, we can take them to intensive care units. And the good thing is, there is an 84% increase in the survival of these people. Yet, the flip side of that coin is, there is also a 12% increase in the disability of these people. Nowadays, we have a lot of people around disabled because they are surviving more. Let me just put that into a context. Right now, at this moment, we're in the place of the world with the highest number of people with a stroke. Let me repeat that. Right now, we're in the second place of the world with the highest number of people with disability due to a stroke. So I'm very happy to be here. I'm very happy to be in this meeting. And let me introduce myself. I'm a physician scientist. I see patients with a stroke. I treat people with a stroke. And my science during the last 25 years or so has been dedicated to study how the brain reacts to the ischemic injury. And my aim is to develop something, a therapeutic strategy to promote recovery in acute ischemic stroke patients. So let's begin with my plan. What is the system I'm working with? It's a very simple system. It's known as the plasminogen activation system. And essentially, there is a zymogen, and that is plasminogen, and that zymogen becomes plasmin in the presence of plasminogen activators. It's a very easy system to describe. Plasmin essentially breaks fibrin into fibrin degradation products, and that's pretty much it. It was discovered back in 1933, and initially it was thought that only bacteria had that system. And the idea was streptococci and staphylococci produce plasmin in order to degrade the extracellular matrix, and by doing so, being able to go underneath the skin and get into the organs. But very soon enough, in 1940-something, it was found that not only bacteria, but also living beings, also animals, have a plasminogen activator. Initially, it was discovered in urine, and that's why it was called urokinase type plasminogen activator, which is the one I'm going to talk to you about. 14 years later, it was found that not only the urine, but also tissues had a plasminogen activator, and to that plasminogen activator was called tissue-type plasminogen activator. The idea was UPA essentially concentrated the production of plasmin on the cell surface, and by doing so, mediated the advance of the cells in the extracellular matrix. And the idea was that PPA was mostly present in endothelial cells, and by being released into the intravascular space, kept the blood vessel open. That was pretty much it. Yet back in 1992 came up this very seminal paper, and I had the original picture of the paper there. That's an cytosymography assay, and what you are seeing there is a rodent brain. The discovery was that those plasminogen activators were not only present in endothelial cells and in urine, but also in the brain outside the blood vessel. And the question that my lab had at that moment, that was 1990-something, was, does that have any relationship with cerebral ischemia? Is there anything related to cerebral ischemia or not? And that's the third point of my presentation today. So to answer that question, we use the model called MCAO, which stands by middle cerebral artery occlusion. What you're going to see here is essentially a mouse, and that mouse is just lying on its back. Let's see if this works. It's lying on its, oh wow, no. It's lying on its back. That mouse is lying on its back, and essentially there is a very long blood vessel going from the neck of the mouse all the way up to the brain. And to that blood vessel, we call it middle cerebral artery. So what we do is we just open the neck, we put a suture into the middle cerebral artery, we advance that suture all the way up to the brain, and we leave that suture into that blood vessel for different periods of time, and then we pull them out. After that, what we get is this. This is an MRI of the brain of those mice. This is the brain in the live animal. What you see in white is the part of the brain that has suffered a stroke. What you see in black is the healthy brain. So the question was what those plasminogen activators were doing in the ischemic brain. What we did was we took a piece of the ischemic brain, the white spot that you see there, we took another piece of the contralateral brain, the non-ischemic brain, and we did something called SDS page seismography assay, which essentially looks like this. With the SDS page seismography assay, what you see essentially is, those are the markers for TPA and UPA. What you see there, how can I point here? Point the pointer here. Okay, what you see here, I is ischemic, this guy here, and C is contralateral, which is the non-ischemic. You see there is a very faint increase in TPA activity right here, very, very mild, and just that very mild increase in TPA activity was enough to produce roughly, this is not working. I'm sorry, but this is not working. Huh? Can I get some help here? No. No, it's not working. There are numbers there. There we go. Okay, thanks. So essentially, since then, up to 900 papers, almost 1,000 papers have been written about the role of TPA in the ischemic brain. Now, you see there is no UPA there. When we look at the brain 72 hours later, at a very late time point, there is no increase in TPA activity, but there is a very significant increase in UPA activity. Way more than the increase in TPA that we saw at earlier time points. The really interesting thing is that despite that significant increase in UPA activity, to this date, only 6 papers have been written on what UPA is doing in the ischemic brain, and that's what I'm going to talk to you about. So this is the UPA-UPAR system. By the way, I have read UPAR in at least 5 of the presentations that we have had so far on repair, which is very interesting to me. So essentially, it's a serine proteinase, which you see there in red, and that serine proteinase binds to this very well-characterized receptor called UPAR, which you see there in green, and UPAR binds to the cell membrane through this GPI anchor tail. The idea is that the binding of UPA to UPAR catalyzes the conversion of plasminogen into plasmin on the cell surface. But it was also discovered that the binding of UPA to UPAR activates intracellular cell signaling pathways. The really interesting thing is, as you can see there, UPAR does not have an intracellular domain. Then it needs a co-receptor to do whatever it's doing, and a little bit more of that later on during my talk. Now, the question is, what the UPA? Okay, what the UPA is doing in the ischemic brain. So the approach that we took was, we essentially struck our mice, and we left the suture there. That's what you are seeing there as an MCAO, middle cerebral artery occlusion, and we probed the brains of those mice to measure the activity of UPA. What we saw was, during the acute phase, there was no increase in UPA activity. Then we returned the animals to the cages. We just pulled the suture out, and we probed their brains at different time points when they were already recovering, and lo and behold, that's when we found a significant increase in UPA activity in the ischemic brain during the recovery phase. The question was as to whether or not the release of UPA at that moment had anything to do with cell survival. So to answer that question, we took wild-type mice and UPA deficient mice, we struck them, and we measured the volume of the ischemic lesion. Very surprisingly, we found that there was no difference. That release of UPA did not have an effect on cell death. To make sure that we were looking at the right thing, essentially we did this thing. We just took wild-type neurons, we put them under OGD, oxygen and glucose deprivation conditions, which I heard today a couple of times. And we measured the medium, just sampling for the release of UPA from neurons. During the acute phase, we just did not find the release of UPA there. Then we return the cells to the incubator, and we just recover them there, and we sample the media again at different time points, and there is when we found a significant increase in the release of UPA. To see whether or not the release of UPA had an effect on cell survival, we just took wild types and UPA deficient neurons, and we did an MTT assay in those neurons after we exposed them to 55 minutes of OGV conditions. MTT assay is essentially to look for neuronal survival. Our surprise was that both strains of neurons essentially died the same. In summary, there is a release of UPA during the recovery phase from an ischemic injury, but that UPA does not have anything to do with cell survival. The question was, well, since that happened in the late phase of the ischemic injury, does it have anything to do with recovery? I mean, what is happening there? Does it have to do any with nerve repair, which is the main topic of this meeting? That was a very difficult question to address, and before getting there, I would like to establish three fundamental premises. Number one is most of the acute ischemic stroke patients experience recovery, not to the extent that they want. 10% have a complete recovery, 10% do not have recovery, and 40% recover to some extent. Number one. That is important because that speaks in favor of the existence of an endogenous system for recovery. And something very, very important for what I'm going to say right now is the recovery after a stroke is driven by structural and functional changes that happen in a very thin layer of tissue that surrounds the necrotic core. Please remember that concept because it's very important for what I'm going to do, okay? So now it comes what animal model of recovery we're going to use. What animal model of neural repair are we going to use? So essentially we took advantage of the forepaw of the mouse. So this is just a drawing of the brain, and this is a picture of the brain after a stroke. What you see there in red is the viable surviving brain. What you see there in white is the part of the brain that has suffered a stroke. So essentially the fingers of the mouse are representing the M1 area, which is right there, which essentially is inside the necrotic core. That's why they lose dexterity after a stroke. Yet the more proximal part of the forepaw is represented in a different part of the brain, which is column 2, which is right there in that part that surrounds the necrotic core, where I just told you recovery takes place. So essentially what we did was we took wild-type mice, UPA deficient mice, and UPAR deficient mice. We struck them, and during 7 days we measured the recovery of the fingerprint of the fingers of the forepaw, the contralateral forepaw. This is what we saw here, okay? What you are seeing there is just days versus strength compared to baseline. What you see on top is essentially sham-operated animals. The surgery doesn't decrease the strength. What you see there in the UPA treated in the wild-type mice, which is the first one, you see there is a drop in strength, and they recover 48 to 72 hours later, at some extent, not 100%. Yet the UPA deficient mice and the UPAR deficient mice do not recover, and that was something very meaningful. What we did next, because we wanted to translate this into the clinic, was we struck these mice and we treated with recombinant UPA through the vein immediately after the stroke. What we observed was that wild-type mice recover a little bit more than they already recovered. That's the curve that you see in the middle. What we saw was UPA deficient mice recover to some extent. What we saw was animals without the receptor, UPAR deficient mice, did not recover. And that was very important because that brought with it this working hypothesis. Number one, UPA promotes neurological recovery after an ischemic stroke. Number two, for that to happen, UPA needs to bind to the receptor UPAR. And the question was, that has to happen again in that very thin layer of tissue surrounding the necrotic core. How can we study that? So to study that, we took the advantage of this technique, DMRI, diffusion magnetic resonance imaging. The basic principle of this technique is the movement of water in the brain reflects the characteristics of the neuroanatomy of that piece of brain. Let me explain it to you a little bit because it's important. Let's pretend that this is the brain. What you have on your left-hand side is the healthy brain. What you have on your right-hand side is the sick brain separated by the green line, okay? I'm talking here about micro-neuroanatomy, just facts, dendrites, synapses, that kind of thing, okay? Let's put a drop of water in the healthy brain. That drop of water is going to move, and to that movement we're going to call mean diffusivity of water, which is very similar across the species, including the human brain. Now, because that water is squeezed, it's going to have a directionality. And to that directionality, we're going to call fractional anisotropy. Closer to 1 means has more directionality, closer to 0 means, because it doesn't have any structure around, essentially it does not have directionality. Let's put the same drop of water now in the ischemic brain. What happens is, because the ischemic brain lost structures, that drop of water is going to move faster, but does not have direction because there is nothing squeezing. In other words, the MD will increase and the FA will decrease. With that in mind, what we did was we took wild type and UPA deficient mice, we struck them, and 24 hours later, we did a diffusion magnetic resonance imaging, and we looked for the MD and FA in that part surrounding the necrotic core when we know the poor part recovers. Essentially, what we found was in UPA deficient mice, the water moves faster, but without directionality. In other words, UPA deficient mice behave, and I'm going to try this thing again, behave like this, like this part right here. That was a very important finding. And that brings us down to the microneuroanatomy. Essentially, the interaction between UPA and UPAR was having an effect on the microneuroanatomy of the ischemic brain, and that's what I'm going to show you to you right now. This is the microneuroanatomy. So essentially, you have a neuron, which is in red, sending signals through the axon, and there is another neuron receiving that information, which I showed you there in blue, receiving that information, and that is the dendrite. The point where the neuron that sends information meets the neuron that receives the information in the synapse, and that's what I'm going to show you here, which I hope is going to work. So, I'm going to need some help. Oh, excuse me. So this is a light confocal microscopy. And what you see there is the axonal terminal right here. What you see here is the dendrite. And what I'm going to do is I'm going to throw UPA. I'm going to add UPA to that system. No, I need to click here. Can I get some help? I need to click here. I need to click in this image. Oh, okay, nice. You could please? Thanks. So I would like for you to take a look at this part right here. This part here, okay? There we go. Here we go with UPA. Just look at this thing. UPA is added, and look at this thing. It moves, and it moves, and it embraces the dendrite, and it keeps moving, and moving, and moving, and moving, and moving. Can we do it again, please? Can we repeat that?
That is sure.
There we go. Here we go. I would like for you to take a look at the tip. The tip is something called the growth cone. The growth cone is a highly dynamic part of the axon that essentially moves, and with that movement, essentially directs the movement of the axon and the recovery of the axon. Now, the question was, where is UPAR there? Where is UPAR in that system? Remember, UPAR is the receptor. So essentially what we took was very young cells, DIV3, and when you take those DIV3 cells, you see UPAR all over the place in all the extensions. That's what you are seeing there in green. In red is just active. Yet, when you take just mature neurons, DIV16, you find that UPAR is present only in those growth cones in a very selective way, just in those growth cones. Essentially, there you are seeing yellow and green is UPAR, the receptor, and red is actin. Let me just show you the drawing. So a growth cone has 3 parts. Has the peripheral part right here, where essentially has filopodia and lamellopodia that move trying to get them to grow. Has a central part right here that has microtubules, and those microtubules just move inside the filopodia and lamellopodia and make the growth cone to grow. And there is a central part with a lot of actin, which essentially acts as a barrier, so those neurofilaments cannot pass. For the cone to grow, essentially what happens is those neurofilaments have to open, and the actin has to open, and the neurofilaments have to get through. That is exactly what we found UPAR. UPAR, as you can see it up here, is very well expressed in the filopodia and in this central area. Now, question is, what is that? What happens during axonal injury? What happened with that receptor? So this was a very difficult thing to accomplish. What we did was we developed this system whereby we culture CNS axons, we culture CNS neurons better, okay? We put it on a drop, on a covered slip, and after 7 days, we just flip it over, and essentially those just send this axonal mantle that you are seeing here. All of these are axons And then we can just get a razor blade and we just can make a cut right here that you can see well. Let me just show you a magnification of that. This is the cut. I hope it's projecting well there. You see, this is one part of the cut, proximal, and this is distal to the cut. And let me show you what happens very soon after the injury. After the injury, you see that new axons are coming up here. They are regrowing. They essentially are regenerating after that kind of injury. Now, where is UPAR there? So we did a staining for tau. Tau essentially stains axons. That's just to show you that those are really axons. Then for UPAR, and let me show you the color picture better, okay? What you are seeing here is all axons here, and you see a lot of UPAR in the tip of those axons after the injury. Let's go over here, and what you see here is, lo and behold, there is a lot of axons here, and the tip of those axons that were injured, there is a lot of UPAR. So clearly, UPAR, the receptor of UPA, was upregulated after injury to those axons. Now the question is, what is UPA doing there? Let's go to the ligand, to UPA, the one that we can use in our patients, in my patients with acute ischemic stroke. So what we did was we took those axonal mantles and we injured them, and we treated them either with vehicle control or with UPA, recombinant UPA. And we did just light confocal microscopy for many hours. I'm going to show you the first 6 hours. So immediately after the cut, what you see is the cut is right here, the cut is right here. Nothing major happens. But essentially within 1 hour, you see axons just growing from the injury side, axons growing from the injury side. And if you look closely, you see that in the UPA-treated axons, the net is more complex, is more imbricated. Now, later on, what you see here is there are axons growing here, but they look kind of wimpy. And the axons growing from the UPA treater look very healthy. They send filopodia. And essentially, what happens later on is in the controls, those axons just go up and come all the way down again. Whereas in the UPA treated, those axons are very healthy. They send filopodia, they send lamellopodia. They look really incredibly healthy right there, okay? So how can we measure that? And this is the data, okay? This is axonal growth after treatment with either vehicle control in white or UPA in black. And as you can see at different time points, which is what you find down in the x-axis, what you see is treatment with UPA increases the growth of those axons very, very quickly. Now, this is going to be a very important piece of information, the one I'm going to give you right now. Let me show you this data in a different way. So essentially the same data, okay? What you see there in the white bars, the first one is at 3 hours, the second one is at 6 hours after the cutting. You see those axons grow a little bit. What you see in the black bars is the growth at 3 and 6 hours when we treat those axons with UPA. So essentially they grow faster. Now what we are going to do is we are going to use a form of UPA that we mutated in a way that that UPA is going to do everything but catalyzing the conversion of plasminogen into plasmin. That's something called ATF because it's the amino terminal fragment of UPA. The surprise was when we treated those axons with ATF, there was axonal growth. In other words, in the brain, UPA is inducing axonal regrowth by a mechanism that does not need the generation of plasmin. And that is very important for one reason I'm going to tell you at the end of my talk. The important thing is when you take UPAR knockout mice, essentially those growth cones you see in the white bars, growth cones grow very, very slowly. When you add UPA to them, they almost do not grow. So UPA needs a receptor to do that. But so far, we are just adding recombinant UPA. What happened with the endogenous UPA? How can we test the hypothesis? So we took advantage of these mice that essentially has endogenous UPA, endogenous UPAR, but endogenous UPA is mutated in a way that does not bind endogenous UPAR. Those are called our ploughed mice. What you see there in white bars, essentially they behave without any sort of treatment very similar to the UPAR knockout because endogenous UPA cannot bind to endogenous UPAR. However, when we treat these mice, we treat these axons with recombinant UPA, what we see is they behave like a wild-type mice. And this is the very first in vivo demonstration that binding of UPA to UPAR, which again, I have seen in at least 5 of your talks before, that binding of UPA to UPAR promotes recovery in axons that have been injured. Now, I'm a clinician. So what is the clinical meaning of that? And I heard the health minister just saying, 5 years to move something from the bench to the bedside. So let me show you the bedside. One of my patients. So this is the brain of one of my patients, okay? The surface of the brain has the cell bodies. And from those cell bodies, axons take from, okay? And essentially, those axons just come all together in this very small, it's like a funnel, very thin structure called internal capsule. The internal capsule is right here and right here, okay? So all the axons converge right there, okay? There are only axons there. And that is a structure called internal capsule. And essentially what you are seeing here is a stroke in the internal capsule, which is very common in people who smoke, who have diabetes, and high blood pressure. It's the commonest cause of a stroke, okay, in those three populations, and in obese people. So essentially what happens is people are left disabled on one side, can move the face, the arm, and the leg in the contralateral side. So we model that stroke in mouse. So essentially what you are seeing there is a coronal cut through the wild-type mouse, and what you are seeing in the square is internal capsule. It's very thin. Let me show you how it looks like. Those green fibers correspond to the internal capsule, are stained for tau. Those are only axons. What we are going to do is we're going to inject endothelin 1 that vasoconstricts that area to produce the same stroke that we are seeing in humans. Lo and behold, we did that, and what we are seeing there in red is UPAR reactivity goes all the way up. So it's the perfect model to mimic in an animal what we see very often in humans, okay? Now let's measure the strength in the contralateral side of this mice. This is going to be a little bit dry, but it's very important, okay? So wild-type mice that you can see there, essentially they dip down, the strength in the contralateral side dips down here, but recovers between 14 and 16 days. They recover very well. UPA deficient mice do not recover well, what you can see there. UPAR knockout mice, which you see down there, they do not recover well. And animals where endogenous UPA to endogenous UPAR don't get together, don't bind to each other, essentially they don't get better. This is very important, has a very important relational relevance for acute ischemic stroke patients. Now, let's treat these guys with recombinant UPA directly into the vein, like we do with our acute ischemic stroke patients. What you can see there, when you treat wild-type mice with recombinant UPA, they get better sooner. They get better 8 to 10 days, like 2 days before that the other animals do. When we treat UPA deficient mice with recombinant UPA, they do better, and that's gigantic. That's really very important. When we treat UPAR knockout mice with recombinant UPA, they never get better. They need the receptor. And again, these are 30 days. We follow these mice day by day for 1 whole month. And the most important thing is when we treat those animals where endogenous UPA doesn't bind to endogenous UPAR with UPA, they get better because essentially we're introducing UPA that does not have the mutation. That is a very important component. Let's look at those brains 1 month later. What I'm going to do is I'm going to get the brain out. That's what you are seeing there. What you are seeing there is a diffusion-weighted MRI. It's the surface of the brain right there. And essentially, in this part where you see the white part is the part of the brain that controls the forepaw, okay? What I'm going to do is I'm going to make the surface of the brain kind of translucent. So you're going to be able to see all the tracks coming from underneath all the way up to the cerebral cortex, exactly the tracks that control the forepaw, the movement of the forepaw, and that's what you are seeing there. This is essentially tracks coming from underneath, and this is when they make it to the cortex right there. That's the part that controls movement. Now, let me show you how a cerebral cortex of a wild-type mice looks like after 1 month of a stroke. The idea is this: wild-type mice, essentially that area is there, but very much reduced. Just compare this one to this one. They recover, but they don't recover as much as the baseline. Let's look at the UPA deficient mice. It's minimal. It's really minimal. And clearly what we are saying here is we're going from way down the axon all the way up to the tracks and all the way up to the connections, which is something, again, that we can explore in humans, and we are kind of parallel between the human and the murine brain. Now, my favorite part, and the last part of my talk, what is the mechanism? So I talked to you at the beginning, and I told you at the beginning that UPAR is bound to the membrane by a GPI tail that doesn't have an intracellular domain, but yet the binding of UPA to UPAR activates intracellular signaling pathways. So it's going to need a co-receptor. So we look for many of those, but the best candidate was this guy, LRP1, the low-density lipoprotein receptor-related protein. It's important because we can intervene that receptor therapeutically, okay? So essentially what happens is it's a member of the LDL receptor gene family, and what LRP does there, which is represented by blue, by purple, is it's like a train. It comes back and forth between the cell membrane and the cytoplasma, between the cell membrane and the cytoplasma, and when it goes to the cell membrane, binds to UPAR, endocytoses UPAR, leaves UPAR down there, and then goes back to the cell membrane, okay? So it's a very interesting thing. So we have a chaperone called the receptor-associated protein, which essentially, when it binds to LRP1, prevents the binding of LRP1 to its ligands. So the question was very simple. If we can block LRP1, we are going to be able to block the endocytosis of UPAR. If we block the endocytosis of UPAR, UPAR is going to last longer on the cell surface. If UPAR lasts longer on the cell surface, then the therapeutic effect of UPA, the recovery repair effect of UPA, is going to last longer, and that is fantastic. And that was essentially the hypothesis. Again, you have UPA, UPAR there, you have LRP1. We're going to interfere with that interaction right here. So UPAR is going to stay there. And as with everything else in science, we were absolutely wrong. When we block LRP1, we did not potentiate the effect of UPA on UPAR. All the way around, we block the effect of UPA on axonal growth, on axonal recovery. This is the same assay of axonal recovery, by the way. We just call them, we treat them with UPA plus minus the blocker of LRP1, which is RAP, okay? So the question was, why? So one thing that we knew is LRP1, this co-receptor, has two MPXY motifs in the intracellular domain that may signal. So the question was as to whether or not that was signaling through LRP1. So we use an antibody that essentially is a monoclonal antibody directed against the last 10 amino acids of the intracellular domain of LRP1 right here. And what we found was that antibody blocked the effect of UPA on axonal recovery. In other words, what we were seeing there was binding of UPA to UPAR was inducing signaling through LRP1, and that signaling was inducing what we were seeing in axonal recovery in the ischemic brain. Now comes the big question. We know that UPAR interacts with integrins. There is a very nice poster about UPA, UPAR, and integrins out there. And we know that one of the integrins that really interacts with is beta integrin. And we know that that interaction happens in some way because it communicates the extracellular matrix with the intracellular compartment. It would be really beautiful to see a system where there is an injury to the brain. The composition of the extracellular matrix changes, and that just kicks the system, and the whole thing just keeps going on. So essentially what we did, we looked for beta-1 integrin on the cell surface. We use a cell surface bioutilization assay, and that's what you are seeing there. And what you are seeing there is when we treat those axons with UPA, UPA, essentially beta-1 integrin is recruited to the membrane. This is, again, a surface bioutilization assay. And the really interesting thing is that recruitment of beta-1 integrin to the surface of the axon cone essentially is mediated by LRP1. If we block LRP1, there is no recruitment. Again and again, the interaction between this UPA, UPAR, and LRP1 was inducing the recruitment of beta-1 integrin to the cell membrane. So far, it was good. And we knew it was important because when we use anti-beta-1 integrin antibodies, which do not induce signaling, when we induce those antibodies, essentially, we block the effect of UPA, UPAR binding on axonal recovery after an injury. Now, the question was, we know that something has to happen in the intracellular part, something right here, because otherwise those axons are not going to grow. What is that something? How can we intervene that from a therapeutic point of view? We look for a number of things. We do proteomics. I have no time to just show you the whole data. I'm going to show you the results. So we found that the binding of UPA to UPAR has a very significant effect on the activation of the ROP family GTPases. I mean, it's very complex, but what we know in the brain is ROP GTPase decrease motility, whereas RAC GTPase increase motility. What we found was essentially that UPA was inducing the activation of RAC1 in those growth cones. We know that that activation was dependent on beta-1 integrin, because if we use beta-1 integrin blocking antibodies, we block UPA-induced RAC1 activation. So far, everything is really getting good. And it's getting even better because when we block RAC1, we block the effect of UPA, UPAR binding on axonal recovery after an injury. So let me put everything together just to conclude, and it's been 35 minutes, which is good. There is cerebral ischemia. And as a consequence of cerebral ischemia, there is axonal injury. As a consequence of that axonal injury, three things happen. Number one, neurons release UPA. It's like the stress response of those neurons, kind of what we were seeing yesterday. There is an increase in UPAR expression, which is essentially the same UPAR that the previous speaker was showing in her list. There was a lot of UPAR in those cells when those cells were placed under hypoxia. And number three, there are changes in the composition of the extracellular matrix. The extracellular matrix is really important in this process. That leads to LRP1-mediated recruitment of the beta-1 integrin into the membrane. Beta-1 integrin induces RAC1 activation, and that leads to axonal recovery. In conclusion, I want to say here that the binding of UPA to UPAR promotes axonal recovery in the ischemic brain. By the way, as a clinician, we already have recombinant UPA. Number two, we know that this effect is mediated by RAC1 activation and is independent of plasmin generation. And why that is important, I told you at the beginning, I was going to tell you why. Because when we treat patients with recombinant UPA, the presence of plasmin induces hemorrhagic transformation in 10% of them. And when they bleed, they have a 60% chance of dying. That is a very bad thing. But now we have the possibility, if this is true, we have the possibility to offer them a form of UPA that does not generate plasmin, and that's my third conclusion, and that therefore does not induce hemorrhagic complications. So I hope I've been able to move this thing from the bench side all the way to the bed of my patient, which is essentially what we want. We're right there in that clinical phase, and I'm really very happy to see how our UPA-UPAR system really integrates with what you were saying before and early today. My most important slide, and I really mean this, is the people that really put up with me, that do the work. As a matter of fact, through the different centers in Emory, I mean, those are the ones that actually work, is not me. This is an MRI of the brain with a CT scan of the brain, human brain, and this was a very strange stroke, has like a question mark, and I use it to say, you have any questions, I'll be very happy to take them. Thanks for being here.
Thank you. My first question, since you mentioned LRP is kind of involved in this process, how about axon recovery in patients or individuals with dyslipidemia, with a high level of low-density lipoproteins?
That is a great question. I mean, if you have dyslipidemia, is LRP going to work there or not? The only data that we have, and we're working on that, is in VLDL knockout mice, but they have hyperlipidemia, you know what I mean? We just don't know yet, but that is a question that we're trying to answer right now. We know that VLDL knockout mice recover in a very different way compared to wild-type mice. They tend to find to have a different type of synaptic recovery, but we just don't know what kind of abnormality is there.
Please questions. Вопросы я переведу, если что. По-русски, по-английски. Okay, I have another question. Why are you always talking about ischemic stroke? What about hemorrhagic stroke? Is some kind of excluded, we're excluding that from your talk. I mean, is it axons are not damaged in hemorrhagic kind of stroke? They don't need to be recovered. Why you limit all of this to ischemic one?
That is a great question. So hemorrhagic stroke has something good and something bad. So when you bleed into the brain, when somebody bleeds into the brain, the hematoma dissects the fibers. Does not destroy the fibers. Essentially, there is an axon here and another axon here, just put them apart.
But this damages axons, dendrite connections.
Yeah, the axons and dendrite just pull away, but when the hematoma reabsorbs, oftentimes they come back together. In ischemic stroke, that doesn't happen. There is a direct injury to the axons and the dendrites, so the type of injury is different. The axonal injury after a hemorrhagic stroke is mostly mediated by thrombin, mediated by PAR1 and PAR2 receptors, and probably plasma. It follows a different pathophysiological event at the ischemic stroke. So that's pretty much.
Yet another question. What makes Russia different from European countries in terms of mortalities caused by ischemic stroke? We have a higher incidence of strokes, or we have a late medical assistance? What's the problem here?
Yeah, for us, you mean why the incidence of a stroke has been decreasing in our system?
No, you show them up, and Russia is kind of a bad situation. What was that?
When we look at that, and it's very interesting, when we look at what is happening in Russia, what is happening here is we see that the 3 risk factors that we have been able to control, mostly all over the place, are very prevalent here. So smoking cigarettes, alcohol intake, hypertension, and dyslipidemia essentially are incredibly prevalent in this area.
No, that partly answers my first question about dyslipidemia.
That is exactly the thing. And the other thing is the acute system to treat these patients is a little bit different here, and that has been evaluated before. So for example, in my system, if somebody comes to our door with an acute ischemic stroke, we have that patient in less than 10 minutes in the angio suite trying to pull the clot out of the brain. That doesn't happen everywhere, and that is a big problem, because that means there is more disabled people in certain parts of the brain. What really caught my attention about that data, if I may say, is the fact that now here is the second place of the world with the largest number of people with disability secondary to a stroke. That's gigantic. That is, I mean, that's why this meeting is great here. Okay, thank you.
Thank you, Manuel, for a great talk. And so we move on.